Good day, everyone. We are very happy to resume our MNS webinars. And today we are very thrilled to have with us Professor Dr. Wael Mohamed. Dr. Wael is a distinguished physician and neuroscientist with the MD and PhD from Pennsylvania State University. He is currently a professor at the Medical School of the International Islamic University of Malaysia. He has delivered over 200 lectures worldwide and published more than 150 peer-reviewed articles. As an editor for several international journals, he has contributed to multiple brain disorder research initiatives, and he has authored around 12 books and 18 book chapters. His research focuses on Parkinson's disease, leading both basic research using zebrafish models and also clinical trials on Parkinson's genetics, especially in the African and Arab region. His team also explores edible bird nests potential benefits and has patented a small capsule for Parkinson's protection. Dr. Wael was recently also appointed as the global advocacy leader for the American Association of Neurology and the World Federation of Neurology. He has secured over $1 million in research funding from different funding organizations, and he's also the founder of the Afrabia Parkinson's Genomic Consortium. Dr. Wael, we are very happy to have you with us today. Thanks a lot. Yeah, and today you are going to talk to us about a very interesting subject indeed. The title is Decoding Parkinson's. The alternative to despair is to build an arc. So we are very happy to have you with us, and the floor is yours.
Okay. Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, good afternoon, and good night for me. Thank you, Dr. Nermeen, for your invitation, and thank you for MNS for this initiative, because it is very, very essential to present neuroscience and different perspectives to our younger colleagues. So today, I mean, my presentation, it is not focused on exploring the data. It is just, thinking in high voice with you about the dilemma of Parkinson's. And even from the title, I selected the titles because so far, I mean, we worked for research to find the cure for Parkinson's. And as you know, there is no cure for Parkinson's, which give us like despair. So we need to maybe think in a different way. Maybe we don't understand our brain. So we are trying to explore together what's the problem and what is the cause of not having a cure so far. So if I ask you to see this picture, comment on this picture, some of you say can see duck, and others can see rabbit, and maybe another group maybe cannot see anything. So it is all different from perspectives, and this is one challenge in Parkinson's, because we don't understand the brain, and even we are doing research, either basic research or clinical research or translational research, in different ways and different perspectives. This is first part of my talk. I will talk about how not to study a disease, and even from the title, you can see you should study a disease. It is like diabetes, hypertension, etc. So it is a disease, and we know what is happening in all these disorders, and we even know how we can control, we can treat. However, in Parkinson's or neurodegenerative disorders in general, or let's say brain disorders in general, we not study disease because we don't understand the disease. So in my talk, we have, as you can see, we have this half. It is pessimistic half. It is dark. It is cloudy, which means we don't. It is a blackout, because we don't have any treatment for Parkinson's so far, which means we are a big failure in treating the Parkinson's. However, in the second half of my talk, so maybe this is the second half, which is flower. It is optimistic one, so we can see. And as you can see, I will not spend the times in explaining what is Parkinson's and epidemiology, etc., because the main problem in the Parkinson's, we have motor symptoms, non-motor symptoms, and even non-motor symptoms, it is they are the most important than motor one, because even depression and anxiety, and some of the patient or many of the patient can execute suicidal attempt due to non-motor symptoms. And even the lay doctors or physicians, neurologists, focusing on motor, because motor, it is what what prevent the patient from normal daily activity, what affects the patient life, and the non-motor symptoms, it is not obvious. And of course, we know Parkinson's, it is a multifactorial disease, which means it has a racial component, genetic component, environmental component, and the comorbidity, etc., and so on. And as you can see, this is we did in a review paper about the Parkinson's and overview in Parkinson's disease in Southeast Asia. And as you can see, a lot of factorials, and it is specific in the area, which means Parkinson's in Southeast Asia, it is totally different from Parkinson's in Africa or Middle Eastern. This is another interesting review, which is 2017. It is 2-century journey of Parkinson's disease research. And if you can, I mean, you have this article, so starting from 1817, and then timelines about the important stations or important invention, important landmarks in the development or understanding of Parkinson's. It stops in 2016. And of course, you can extend the line until 2024. However, as I said, the main problem here, for decades, some of our best and the brightest medical scientists have dedicated themselves to finding a cure for neurodegenerative and Parkinson's especially. So what's happened? What is the cure? And this is the main dilemma. And of course, it is a big problem for Parkinson's in developing countries. We cannot, I mean, enter in much more details. This is Michael Fox, and of course, we knew him from Back to the Future, and he had Parkinson's, and it was like changing his life, and he established Michael Fox Foundations, and it is a very big foundations in the US and worldwide can promote for discovery of the treatment for the Parkinson's. So I met with him once in the US. He is a very enthusiastic person and a dedicated person. This is one of his books. He has several books when it comes living with uncertainty. I mean, it is like no time like the futures. The actor and activist opens up about his newfound equally uphill brand of pessimism, which means uncertainty. What is uncertainty? Because the main issue, one of the main issue in the treatment of Parkinson's, when you have a patient in a clinic and you decided or you realized or you know he or she has Parkinson's, how you can break out the bad news to the patient? Because if he or she is an intelligent or highly educated patient, he knew that there is no curative treatment, which means this is the end of the career. And as we knew about Robin Williams was diagnosed as a Parkinson's, or some others say it is frontotemporal dementia, which is in the same track. And maybe 2 months after the diagnosis, he committed suicide. And one explanation, it is, I mean, he knew that there is no cure, and which means this can destroy his career, etc., and so on. And also, if you know, this is a Deadpool. He is Ryan Reynolds. I mean, his father is one of the Parkinson's patients, and he is one of the trustees and board members in Michael J. Fox. And also, he has a famous interview about his struggle with his mother for taking care of his father. And the main problem he faced with his father was like non-motor symptoms. It is not motor symptoms. And a third one, it is he is NFL Brett Favre. He is a famous football player in the US, and also he got Parkinson's, and this destroyed his career and making a lot of financial problems to him. So now the main questions, we have two main questions. Do we really understand the Parkinson's? Because technically we don't have cure, which means we don't understand the disease. This is one of the explanation. How far have we come and how far can we go further? And of course, if you are a clinician, you meet with the public, or you meet in the maybe interviews and on the mainstream or podcast or whatever. And the public always ask you, I mean, where is our cure? What happened to our cure? Because there is no cure. And they have justification. They can say we bear the costs of this disease, both financial and emotional, because actually most of the organization in the research, it is from taxpayers, so they pay for research grants that we are using to examine the Parkinson's, and yet we failed or we let them down because we don't have any cure. And they can also say we have invested heavily in this research, both clinically and basic, and many patients, Parkinson's patients, voluntarily can participate in the clinical trials, etc. And this is for the hope of the disease. We have steadfastly supported this research with our tax dollars and with our bodies as volunteers in clinical trials. We have been patient, but after all these years, we have the right to ask, where is our cure? And even at the level of the government or countries, this is during Obama era. This was famous quote for Obama, from Obama about on the brain initiative. As humans, we can identify galaxies light years away, and we can study particles smaller than an atom, but we still haven't unlocked the mystery of the 3 pounds of matter that sit between our ears, which is very, very true, because to cure brain disease, we need to first understand how our brain works. And even in 2016, and the Obama administrative signed was very optimistic, bills about the Parkinson's, and they claim that they will use, genetic therapy, biotechnology, nanotechnology, etc., to find the cures. And now Obama, gone, and there is no cure so far. And even with Biden, he signed specifically a national plan to end the Parkinson's Act, a law that will help prevent, treat, cure Parkinson's and similar disorders. This law is about dignity. It gives people hope that we can end this cruel disease, and that can still do big things. And again, it was, a long time ago, 2023, I think, or 24. So, so far there is no cure. And recently, Michael Fox got the Medal of Freedom from Biden before he left the office, and this is for his work and his enthusiasm and find the treatment of the Parkinson's. And even globally, we have 2 major projects, which is Brain Initiative and Human Genome Project. And there was like very large amount of money, but we didn't have any cure so far. And as you can see, there are tremendous increase in publication in neuroscience, either basic, clinical, or translational, but we didn't have any cure yet. And this is some of the newspaper that explain or trying to explore why these projects, big mega projects, multi-center, multi-billions project, fail to understand the brain. The Human Brain Project hasn't lived up to its promise. And as you can see, 2013 and until 2023, and as you can see, this is the number in funding, which is a huge funding. Why the Human Brain Project went wrong and how to fix it? One possibility is poor management, or maybe it is a fundamentally wrong with big science, which means big science cannot explain how our brain works. So, which means we don't understand how brain works. So to conclude, for this part, there isn't a single agreed upon theory of how the brain works. And not everyone in the field agreed that building a simulated brain was the best way to study it. And this can explain so far why we failed to understand our brain. At some point, we thought that genetics is an answer. So you can see a lot of publications about the genetics cracking of the code of the genes, and we can understand whatever. However, it turns that genetics or genome is only one piece of the large puzzle, and we examine exosome, epigenome, microbiome, all omes. So and again, it is still mysterious to us how brain works. Which means we moved from DNA to the human as a human, and also we moved from the human to DNA. So we examine DNA, chromatin, RNA, protein, metabolite, pathways, cells, circuits, and so on. However, we don't understand the brain, and we don't have the cure. So far, we have not been able to translate new knowledge into a new and effective therapies. This is maybe due to complexity of our brain, maybe complexity of the biology of our brain, maybe this is due to disease heterogeneity, because as you know, BD is a multifactorial and a lot of heterogeneity, a lot of factors. So we cannot simulate what is happening in human using animal model. Preclinical models are not predictive, which means we don't have so far any animal model that capsulates, that can express all what is happening in the real life for BD patients. Also, reproducibility problem, maybe drug discovery, and this is one layer of complexity again, because most of big pharma companies are reluctant to invest in investigating the new cure for neurodegenerative disorders, because there is no big revenue. And if you remember, during the COVID era, so COVID vaccine, it is highly rewarding for the pharma company. So the opposite is for neurodegenerative in general and the Parkinson's. The clinical trials, it is difficult and costly, which we can say the reductionist approach has reached its limit. And as I said, we moved from genes to proteins to cells to circuits to behavior, and in the opposite direction. I will end this part with a common or old example. It is a duck of Vaucanson. In France, it is in 1764, one engineer called Vaucanson, he invented digestive duck. He wanted to imitate the nature duck, or what is happening in the normal duck or natural duck. So he invented metallic duck and try to mimic what is happening in the normal duct in terms of this metallic duct. Vaucanson duck or digesting duck can eat, so can put food in the mouth, so can eat, and also can excrete, defecate, which is can do. However, he failed to make the biological link between mouth and defecations. What is happening here? So this model failed to mimic what is happening. So it looks like a duck. So it is a duck because it can walk, it can have sounds, it can eat, it can defecate. But it is not a real duck, because in a sense of there is no link between eating and defecation. This is the metaphor, so can reflect on what is happening in Parkinson's. Parkinson's, it is a disease. It looks like any normal disease, but it is not like normal disease, because we don't understand the complexity, so we don't have any treatment. So I put the comparison between that of Vaucanson and the current understanding of Parkinson's. So in terms of appearance versus reality, models, which is animal models, mimic symptoms, but fails to fully replicate what is the disease complexity, what is happening in human, which means by end of the day, if I am working on rats, shrimp, or zebrafish, I couldn't extrapolate my data to the human setting, which means by end of the day, we are not interested in treating Parkinson's in rats because we need to treat Parkinson's in human. Mechanistic illusion focus on isolated mechanism without addressing the disease systemic nature, reductionism, and the limitations, aspiration, and outcome. So this is just to maybe to a way to understand why we failed to have a treatment. And again, this is, for example, agent used in Parkinson's clinical trials by blind in different phases of drug development. And it is all about symptomatic treatment. It is disease modifying. However, there is no curative treatment. And if we take one big company like Pfizer, it is in the business for 175 years and has not cured one single disease. For me, maybe this is some behind the scene issues. I don't know, but by end of the day, we don't have any big investment from Pfizer in the Parkinson's disease. They tried several modalities of therapeutic modalities like oral injectable drugs, gene therapy, cell replacement therapy. So, however, again, there is no curative treatment. Maybe this is due to inability to detect the disease before it has progressed. This is another layer of complexity in Parkinson's, delayed diagnosis, because even a lot of issues like sociocultural issues in diagnosis. Biogene and Sage to scrap neurological disorders, drug development after trial failure. And this is in 2024. And again, this explains why most of the big pharma companies are not investing in finding cure. Clinical trial failures, financial burden, complex disease mechanism, lengthy regulatory processes, and the limited market incentive, as we explained. So this is for the first part, which is the pessimistic part, which is the dark side of my talk, which means, I mean, we supposed to do what we do as a scientist, researchers, physician, etc. This is for sake of our patient and humanity to find the treatment. So one of the maybe method to thinking outside the box is using the right animal model. So of course, before I went to USA, I worked as a neurologist, neuropsychiatrist. So I worked with a patient, and then in US I worked with rats and mice. And when I came here in Malaysia, so I challenged my comfort zone and I started zebrafish. So zebrafish may be the arc to save our patients. So animal model of neurodegenerative disease, we have challenges like recapitulate only a subset of the disease pathology and the phenotype in humans. As I explained, it is the heterogeneity is fast. Drugs that are safe and effective in animal models are not safe or as effective as in humans, and also very short lifespan. You know, if we compare between rats, mice, zebrafish, etc., there is a short lifespan, and we need to mimic what is happening in human, because technically most of the neurodegenerative, it is old age, maybe after 40, after 50. So we are eager to introduce the model. However, we are trying to push the biological processes to catch the chronological age of the animal model to be coincident or corresponding to what's happening in human, and this pushing can interfere or confound factors in our result. So again, this is difficult to extrapolate data from animal to the human, which means this is what I explained. If you compare me and the mouse, so technically we are the same, but the physiology, metabolism, immunity are totally different, which means we are trying to push lesions to try to speed up the processes, because mice only live 2 years. But by pushing it, that does not necessarily mean that the processes is the same. So this is one of the confounding factors, and we cannot extrapolate our data to the human. Come to the zebrafish, it is up to date now, and it is a very important animal model for studying many diseases. And by 2035, US EBA will eliminate all mammals testing, which maybe we will be obliged to use zebrafish instead of rats. And I will not enter into the advantage and why we select zebrafish as a model, because this is not I mean the focus. But in general, these are the perfect choice of the zebrafish in Parkinson's disease. And the main challenge, some maybe any every one of us, I mean, has like comfort zone working with rats, with mice. So there is a challenge to challenge your comfort zone and switch to the zebrafish. We are not familiar, and even in Africa and Arab borders, we are not familiar with zebrafish. However, it is very affordable, and there is no need for extra money for breeding. This is what we designed here in Malaysia since I came. Zebrafish housing. This is the front, and there is a already ready-made setting, which is very expensive, but we came up with this setting, which is handmade with local materials, so it is not a very expensive, and it is easy to manage. So I will focus now on zebrafish BD models. We can make neurodegenerative models using zebrafish in Parkinson's, Huntington, and Alzheimer's. And in Parkinson's, we have three main ways. We here focused on neurotoxin because it is much more cheaper and it is straightforward. And we can use three toxins, and each one has pros and cons, advantage, disadvantage, but we used MBTB. And in zebrafish, we can have all behavioral testing that we can have in mice, in rodent, and others, like novel tank, mirror, object discrimination, showing behavior, social, male interactions, and learning avoidance, and all other tests. One important technique is anesthesia. How to can make anesthesia? We can make anesthesia in two different ways. Injections, intraperitoneal or in ice cold, and it is totally different from male and female. So we need to know these techniques. And in this video, we have this video. This is one of the anesthesia. It is intraperitoneal injection. Okay, and in the second one, or the opposite one, it is anesthesia using ice cold method. So either one, it is effective, and we can use it. Okay, this is for anesthesia. For behavioral analysis, we have like big chart can explain what we can comment on the locomotor activity and swimming pattern, which is behavioral testing. And of course, we have certain parameters like freezing bouts, frequency, time, turning angle, and so on. This is a small, very primitive setting that designed. This is a camera, and this is a box, and this is another box, and this is software. The main challenge here is software, but we managed to have it. This is some videos I will share with you. This is a normal fish, and as you can see, I mean, it is relaxing. In this video, I will share a series of videos, which means normal and then MBTB in different ways. So I can start.
This is very interesting.
Thank you. And of course, we have, this is the tracking videos, and it make visualization of the track the direction of the zebrafish. I mean, it is totally different from normal. This one, it is and MBTB, and we can analyze, I mean, different parameters. This is why we use the MBT model, because we can mimic what is happening in humans. So in human, for example, in Parkinson's, in real patient, we have bradykinesia, we have rigidity, tremors, postural instability, gait abnormalities, hypokinesia, and freezing movement. And this is, we came up with glossary, which is very interesting, and we shared with other groups, which, what does it mean, bradykinesia in zebrafish? It is significant reduction in swimming speed and impaired spontaneous locomotion. What is rigidity in zebrafish? It changes in muscle tone and stiffness. Tremors, what is it is abnormal oscillatory swimming pattern. Postural instability, impaired balance, coordination can be indirectly assessed. And of course, we are trying to update. Sometimes we have like bizarre behavior and so on. But anyway, the basic component or the basic symptoms and phenotyping of Parkinson's, it is established in Parkinson's in zebrafish. Also, we compare between zebrafish and the human, and this is the relation. In terms of motor symptoms in general, zebrafish motor deficits mimic human bradykinesia and gait issues. Toxins induce similar dysfunctions. Loss of dopaminergic neurons. Zebrafish dopaminergic neurons are functionally analogous to human neurons. Toxins, genetic models replicate neurogeneration. Alpha-synuclein aggregation. Zebrafish model help study alpha-synuclein, etc. Non-motor symptoms, even non-motor symptoms. We can examine other motor symptoms. It is like in human. Oxidative stress, it is also shared mechanism of oxidative stress between human and zebrafish. Response to L-DOPA, mitochondrial dysfunction, or factory deficit. In olfactory deficit or factory dysfunction mirror human hyposmia, which is one of the, again, important non-motor symptoms of Parkinson's disease. And it is now it is established as an early sign and symptoms of Parkinson's. Cognitive decline, also we can mimic. So in general, zebrafish has many advantage in using in Parkinson's disease modeling, conserved pathways. Zebrafish share key molecular and cellular mechanisms with humans, which is dopaminergic and alpha-synuclein. Symptoms overlap. Both species exhibit motor, non-motor symptoms, enabling holistic study, which is very important. Translational utility. Zebrafish models are used to screen therapies, which is very important aspect of using zebrafish, and study disease progression. Of course, it has limitations. One of the limitations, it is substantia nigra. There is no substantia nigra. However, there is posterior tuberculum, which is functionally analogous to substantia nigra. It is same dopaminergic anatomy. And of course, zebrafish doesn't have lung, so some limitation. So again, I will share another set of videos. Again, this one is normal. This one is MBTB. And of course, in MBTB, we came up with a specific protocol for dosing and the protocol and how you can inject MBTB, because again, symptoms or phenotyping of zebrafish depends on the dosing of MBTB and how long after the injection of MBTB. Again, this is the tracking, and we can explain this. Erratic movements in general, it is what we called in human, it is abnormal movement like myoclonus, like Abuelezz, like Chorea. We can try to find it. Again, this is normal one. This is myoclonus. Myoclonus in human, it is myoclonus. It is colonus. It is twitching muscle spasm without any relaxation. Here it is jerking and twitching of the fish, and as you can see. Another symptom, which is Abuelezz, which is in Abuelezz, and it is common in Parkinson's in advanced cases. Here it is violent fringing of extremities, and as you can see, this is what similar to Abuelezz. Chorea, slow twisting or continued movement. Of course, we have different types of chorea, and even hemichorea and so on. So we can find different modalities. It depends on the dosing, how much you inject of MBTB. which means how much you destroy, how much dopaminergic centers you destroy. Is it complete destructions? Is it partial, and so on. Another chorea. This one normal. This chorea, slow twisting or continued movement. Of course, I mean, this is for memory and learning, 3-axis maze, and we designed this from plexiglass. It is very straightforward, and the original one, it is very expensive. So we came up with this one. So it is classical maze, but this is for zebrafish. And of course, we have different parameters, and you can feed into the software, and you can have, I mean, complete analysis. This is how we take the zebrafish brain, harvest zebrafish brain. Okay, so finally, I will share. This is the collective video. We had a small team here, and again, it is the setting of the lab. It is very small and also not very expensive. Okay, so as the final part, I want to maybe talk one slides about the smart capsule. So now we divided our work into different phases. Phase one, it is mechanistic study, which means we need to understand what is happening in Parkinson's. As we explained, maybe we don't understand. So we spent maybe 5 years now or 4 years and a half to understand what is the mechanism, and we focused on HMGB1 complex, which is very important. So we are trying now to work or play with HMGB1 to have like improve or cure for the Parkinson's. We cannot claim we have cure, but it is a humble trial from us. So we used the nanotechnology to make smart capsule, which means it is a capsule formulated with nanotechnology, and it can cross the blood-brain barrier very easily, so can deliver the drug and the herb. We have herb here. It is called edible bird nest. For edible bird nest, it is a common herb in Chinese medicine. It is very common in Southeast Asia, and it is very expensive one because it is very rare. It is widely studied in many diseases like cancer, diabetes, hypertension, etc. However, we are the first group to study in Parkinson's exactly. It has like many postulated mechanisms in Parkinson's. However, we are focusing on HMGB1. So it is like working with microglia. It is working on oxidative stress, etc., and so on, and it working helping in motor functions and the learning and the memory, neuroinflammations, oxidative stress, and apoptosis. We had the preliminary good result and the promising one. So in the capsule we have extract of edible bird nest and inject it into the zebrafish, and also we can add some anti-Parkinson's drugs, antidopaminergic like dipranil, for example. So it is under revision for the patent. It may take 1 year to be decided and to be enter into another phase, which is clinical trials. So I will conclude with few slides about what we discussed now. So zebrafish, as a zebrafish, it is a great potential and alternative for vertebrate model. Maybe it is an arc. It is it is a one way to understand better Parkinson's. Becoming the model of choice in different ways. Many commercial companies now have brought zebrafish into their portfolio, which means if you apply for any grant to zebrafish model, this is very prestigious or up to date. Despite limitations, zebrafish can effectively complement other mammalian and non-mammalian models. So again, we aimed for the moon and got the stars. You miss 100% of the shots you don't take. Perhaps we did hit the right targets, which means missing the marks is one of the ways in which we learn to hit the target. So in my presentation, it is not like calling for to be frustrated and desperate. However, we are trying much more to maybe think in a different ways. And one important feature of zebrafish, it is the ability of zebrafish to regenerate. Of course, this is very famous or very well known in the heart. Heart can regenerate. So it is very widely approved. However, in nervous system, it is also there is regeneration power of zebrafish. However, it is under investigation. And this is again quote from Michael Fox: acceptance does not mean resignation. Acceptance of BD is a very serious problems, and we cannot solve it. It is not resignation. It means understanding that something is what it is, and there has got to be a way through it, and this is very inspiring from Michael. And again, we failed as I explained. However, this explained the failure could be like fall, acknowledge, investigate, learn, understand, realign, and involve. And this is important. Chinese, it is in Chinese novel, which is very important. If you read it, of course, there is an English translation, How to Get There. So in How to Get There, if what we need in Parkinson's, we need to get there. There it is means finding the cure. So go to the end of the path until you get to the gate. So this is explanation. So go through the gate, and we went through the gate. Keep going towards the horizon. We keep going. We keep investigating, trying basic and clinical. Sit down and have rest every now and again, because now we don't have cure. So we are exhausted, and even we are under ultimate pressure. Even if you get money from the grant, you are under ultimate pressures and tensions to find, maybe positive result, maybe something new. You cannot replicate what others did, but keep on going. Just keep on with it. Keep on going as far as you can. That is how you get there. So we need to keep going as far as we can in terms of using animal models, translational and clinical. And of course, this is interdisciplinary, and so we need to work together. This is our team, and as you can see, we have in molecular persons, we have zebrafish expert and also some behaviorist, neuro behaviorist. We got funded from national, from Malaysia here, from FRGS, PRGS is from Malaysia, and also got funded from international funding. Thank you for having me, and if you have any questions.
Thanks a lot. Thanks a lot, Dr. Wael, for this interesting topic indeed. And I'm sure that, it raises some questions. So if anyone of the audience wants to ask, please feel free to unmute yourself and just ask your questions, or you can send it in the chat. Okay, Dr. Wael, I have some question. What kind of non-motor symptoms you can observe in zebrafish?
You can have like dementia. We can examine learning and memory. We can examine epilepsy, like seizures. Sometimes it is seizures. Depression and anxiety. It is model for zebrafish. It is established. There is established model for depression and anxiety. They're freezing, like epilepsy, like seizures. This is for what we explain.
Yeah, and concerning Parkinson's, the non-motor symptoms in zebrafish, it can be tested.
Yes. It is. We can have zebrafish model of seizures, for example. Okay, so we introduce Parkinson's and then introduce seizures in zebrafish and see how it works. Characterize onset, offset, what is increased, what is decreased, and again we can add the dementia, we can add examine learning and the memory. But I mean, this is very very expensive or very complex behavior. So we are trying to move in phases. So now we are in phase 3, focusing on, as I said, the smart capsule, which is a therapeutic. So we did mechanistic, then now we are in therapeutic phase.
Yeah. Another question: does do the animal behavior testing in zebrafish can be verified using animes? For example, a software, well-known software like animes or
yes,
thing,
there
or
is we are using Danio Vision, which is I think you can use at any model, because there is there is items you can analyze. And as I said, you put the items, you explain the items, and then the software can do the rest. It is straightforward. There is nothing. The main point is how to put the data into clinical setting, how to explain what you are seeing in zebrafish in clinical setting.
Yeah. Another question: how can we obtain this model in Arab countries like Egypt or Jordan? Maybe. Do you know if any facility using it?
I think in Egypt, no, there is no zebrafish. I tried several times in Egypt, but I mean, Arab people do not collaborate.
It takes a lot of time it takes a lot of time to develop something.
We are trying now in Africa. So in DRC, we are starting Ghana. We started, and even we applied for a big grant for the Belgium. So they will send the setting, the ready-made setting. In South Africa, they have good facility, but they are working in cancer and diabetes or something like this. So it is easy. Could be if anyone is willing, we can collaborate. And even we are in contact with the Japanese group who can make knockout morpholino. This is the genetics that I mean. We are not there because they're expensive. They can give us the model for free, but we pay for shipment. Here it is okay, it is easy. But I think in Arab countries, Egypt, for example, it is very difficult to make exportation or importation, because security check, etc., and so on. Security meeting. I think this is the case. But here we got the
Yeah, this concerns the transgenic model only, or is it affordable and easy introduced to introduce zebrafish into Arab countries like Egypt with an affordable facility or something?
No, I mean you can use you know the market zebrafish. You know zebrafish in you know if you go to the I mean Bits shop, you can find zebrafish. It is okay, but again it is not the same you know race. We used the one from Japanese, which is the original one.
Yeah.
To import, I mean it will you know a lot of you know
Yeah,
signatures.
the hassle with the importation and yeah.
Yes, I mean, if you have the way, if you can overcome this, I mean, we can work together. I mean, it is not a big deal.
Yeah.
The big deal, it is again, it is in dealing with security and approval.
Yeah. Another question about the smart capsule. Is it effective for all forms of Parkinson's? So have you tried it on familial Parkinson's or different types of mutations? Or
So just,
to us?
we cannot claim it is curative. Okay, what we you know what we say here, conservative result or conservative proof, which means we tried in zebrafish. There is a neuroprotection. There is a curative in zebrafish, and it should be tried in rodent like rats, mice, then monkeys, and then go to clinical trials. So it is a long step, long way, but we can claim that our targets is to pass blood-brain barrier, past the blood-brain barrier, to deliver the drugs or edible birdnest or herbal into the high concentration inside the brain, which is specifically substantia nigra, you know, striatum, etc., and so on. It did. It treated, yes, it treated, but again, we cannot claim this is a curative. It is long way. But even, I mean, if we have more money to make more investigation, we moved because we introduced the model, we evaluate the model behaviorally, histopathologically, genetically, okay, and then we interfere or we trying to treat with smart capsule. We did improvement in alleviating the symptoms. We submitted to the Malaysian patent, which means if approved, this means the smart capsule will be approved in Malaysia. It is not international. You need to move to the international. It is a long way, so who knows? I mean,
of course. Yeah, but the preliminary studies are very promising indeed, and they need maybe further improvements or further expansions to other models. Yeah.
I mean, we are collaborating with Prof. Azazi in AUC. So because there is a technique in nanotechnology, BBE or something like this. This is a very you know new one. And of course, I am not in biotechnology. But
it's more for the brain targeting, maybe to make it more brain
this
specific.
is the main idea. So the main idea.
Yeah.
And even it the name Smart Capsule. This is for you know commercialization, but in scientifically we didn't say smart capsule because I mean it can interfere with lot, but this is for you know just publication.
Yeah, I see. So good luck with the work, and yeah, it seems very interesting.
Thank you.
Any further questions?
I don't have any question. I just wanted to thank you, Christina Dalla, the president of the society. I'm sorry I had some technical problems with the camera and the signal, but I heard your talk. It was amazing, and thank you for your support and your work. And thanks everyone for being here today.
Yeah, Professor Christina is the president of the MNS Society, and yeah, she's having a little bit of technical problems. But she's thanking you very much, and she said that the webinar was very amazing, and thanks a lot for
I mean, we are ready. We have open heart, open chest, open mind, so ready for any collaboration. And of course, Dr. Amira, I worked with Dr. Amira before. Mark also, I worked with Mark, and I mean, we are ready to collaborate. And as I explained, I mean, we have much more details about our zebrafish using and also in clinical trials, because we have consortium to studying the Parkinson's in clinical trials, working with Southeast Asia, like Thailand, Philippines, and so on. So we are welcome. Just you can shoot me an email. We can start brainstorming, apply for any grant or make any collaboration. Thank you.
Great, thank you. That's impressive. Thank you very much.
Thank you.
Thank you.
Thanks a lot, Dr. Wael, and it has been a pleasure having you with us today. And we hope that it won't be the last one. Looking forward for more collaborations. And Christina, do you have to add anything?
No, that's all. Thank you very much, and we hope we'll see you also at some of our meetings.
Yeah, of course.
Yeah,
Okay, thank you. Maybe you can visit us in Malaysia or Egypt. I mean, we have 2 countries now, Malaysia or Egypt. So thank you so much.
Thanks a lot.
Thanks
a lot. Thanks a lot, Dr. Wael, and thanks a lot for all the audience. And
okay, thank you. Okay,
thanks.
bye-bye.
Thanks. Bye.